Well, welcome everybody. It's good to have many people on again. My name is Jack Dekkers. I'm a faculty member in the Department of Animal Science at Iowa State University, and I'm one of the co-PIs on the Agricultural Genome to Phenome Initiative funded by NIFA. It's time for our 26th field day, and very happy to learn about a vision for more sustainable integrated crop and livestock production systems. And we're going to have two presenters, Dr. Ed Buckler, who is a research geneticist at USDA ARS, and also an adjunct professor in plant breeding and genetics at Cornell University. He'll be our first speaker. And then he will be followed by Dr. Mark Bogus, who is the director of the U.S. Meat Animal Research Center in Clay Center, Nebraska. So two speakers from ARS, and ARS is a partner organization for this initiative. And so we're very pleased that they're able to contribute and they have been contributing all along. But more specifically hear about their thoughts that they have for a circular economy approach for reducing nitrogen use inefficiencies in integrated livestock production systems. So with that, I'm going to turn it over to Ed.
Okay. Well, Jack, thanks very much for the introduction. And yeah, let me get started here. And so, yeah, one way, our effort here has been focused really on the whole thinking about how does nitrogen move through the system. And, Mark and I will be presenting it, but I know we've talked to hundreds of various people to think through this problem, talk through it. And, what we're distilling here is really some of those ideas that are coming from all different aspects of agriculture. So agriculture has a nitrogen problem. Obviously it's really important. It's how, of course, we are able to live is by agriculture providing us that nitrogen. But together across the system, it's producing about 11% of total US greenhouse gas emissions. And, some of that is essentially, the CO2 coming from the Haber-Bosch. It's the nitrous oxide that is being emitted from our soils that we put after we put fertilizer down to supply our crops. It's methane that is coming from the livestock production, and it's also from manure coming off the system. And so we'll start out here with a question for everybody as to which of these do you think is the biggest contributor to US greenhouse gas emissions among agriculture? Please vote away. And then Nicole will share with us the results. Nicole, do you want to share results?
Yes, they should be up there. If you click on the polls at the bottom, it should pop up on your screen.
Okay. Sounds good. So is it popped up now or no?
No, but I can tell you most people said nitrous oxide released from the soil.
Okay.
That's the popular answer.
Okay. So by the way, we have a very informed group of people here, because when I talk about this to a wider audience, the vast majority of people will pick out enteric methane is the #1. And that is true globally. That globally cattle methane is a larger issue than nitrous oxide. But in many regards, it's because US agriculture has worked so hard over the last several decades to become as efficient as it possibly can, that really now the biggest problem, followed by enteric methane, is nitrous oxide coming off the soil. So it's a sign of our success in agriculture that it's nitrous oxide is our biggest problem here. Okay. And so in a lot of other areas and across the US economy, we do have some strategies now starting to be implemented to reduce greenhouse gas emissions. And so if you look at, whether that's electrification of our power system, vehicles, and so on, we're now kind of on target in the US to be following this blue line in terms of greenhouse gas emissions. And maybe not by 2050, but by 2055 or 2060, we should be on that target. What you'll see, though, is the current projections for agriculture has the agriculture is getting substantially more efficient over that time, but not really reducing absolute levels of greenhouse gas emissions. And so that puts agriculture by mid-century being responsible for about nearly 40% of US greenhouse gas emissions. And I think that none of us want to be in a sector where we're producing nearly half of US greenhouse gas emissions. And so I think the question is, are there ways we can shift this and put us into better balance? And so a group of us across ARS, including Lisa Ainsworth, Mark, Virginia Jin, Peter Kleiman, and myself, started working across sectors to ask the question, how could we shift this? And so a lot of it has to do with essentially, we have these nitrogen inputs, whether they're synthetic fertilizer or natural fixation, but 87% of them are getting lost. So about 70% of that's going into the atmosphere. And some of that is nitrous oxide, which has this global greenhouse gas warming of about 300 times of what CO2 does. And then we also have lots of that is about 17% is leaching into the water and is producing a lot of water pollution throughout the system. And so obviously that's really inefficient to have done all this effort to fix the nitrogen in either approach, and we're only getting 13% of it through to the consumer. And so the question is, how can we address this? And I think in the last decade, there's been a lot of pushback against synthetic nitrogen. And what we would argue is that really Haber-Bosch itself is not the problem. And people, writers and thinkers in this area, for example, Václav Smil has made this very clear that Haber-Bosch is much more land and time efficient. By mid-century, 2/3 of the globe is likely to be dependent upon it. And from one perspective, people have highlighted that it uses about 1% of global energy. But I think another way to think about that, essentially, we only use 1% of global energy in order to fix something that is absolutely critical for the entire food system. So I don't think really synthetic nitrogen or Haber-Bosch reaction isn't really the problem. It's how we use that synthetic nitrogen in our agricultural system. And a lot of the issues have to do with the trophic levels of moving nitrogen through each of these different steps before it gets to us. And so the soil, and in the case of the US right now, we have soil and microbes are taking it up. Plants, we're losing about 50% of the nitrogen right there. Those plants are then producing this crude plant protein and DDGs. And the problem with that crude plant protein is it's not as absorbed as easily or as efficiently as things like amino acids, which are those are absorbed about 2 to 3-fold more efficiently than that crude plant protein. And so that you combine those two losses together, you're now at 1/4 to 1/6 of the efficiency than if you were to directly provide, for example, amino acids to livestock. And so this approach that we've kind of taken in this is to say, how can we shift this the nitrogen in the system? And the first is to essentially use that Haber-Bosch nitrogen and feed our livestock, in the case of cattle, things like urea and non-protein nitrogen, much more of it should come directly from those synthetic sources rather than coming indirectly through plants. We also can use that directly in amino acid synthesis, which of course we already do amino acid synthesis, but we're talking about doing this at a much higher level and providing a substantial amount of those amino acids directly to our non-ruminants. And those amino acids can also go into alternative and cultured protein production systems. So, it would be a shift of being a few percent of our production right now to perhaps even half of the total flow of nitrogen would come out of those sources. And then the key thing that allows us to do is on the cropping system, it allows us to change things so that we can essentially recycle nitrogen on the cropping side. Because right now we're using so much of the cropping system to pull nitrogen off those fields to provide livestock their protein. But if we can recycle that nitrogen, then we can have a much more sustainable system on the cropping system side. And so we think together, these allow us to make a much more circular economy. And if you're doing that, then you need to put less fertilizer on the ground, and with higher levels of amino acids, we should also have substantially less manure problem. So instead of the current approach here, we're certainly going to still apply fertilizer, but we're producing less crude plant protein, and we complement that by things like more amino acids and urea, essentially really a targeted nitrogen use at a higher trophic level. And so I think together, we think these types of approaches by mid-century could reduce agricultural greenhouse gases by 50% or more. And so what would it take to implement this? First off, there's no one invention or discovery to shift a big agricultural system like we have here in the US. It's going to require shifts all across the system to make this happen, discoveries in science and engineering to make that happen. And they cover everything from in a lot of these different areas. And I'll dive into a couple of these just to give you a feel for it. And then later on in the second part of my talk, I'll talk about some of the areas we're working on the corn side, and then Mark will pick it up on the livestock production side about where this can go. I think the first thing we really need in this whole area is there's a lot of opportunities for modeling. We need to have much better models of how nitrogen is flowing all the way from soil through the entire ecosystem. The modeling at the economic and trade impact, a lot of the big trading that goes on in agricultural commodities is really about moving nitrogen around the globe. Then how is climate and human health impact going to affect these? And then what are exactly the policies that could really accelerate this type of transition? I think there's lots of policy ideas that could help make this type of shifts, but we need that type of modeling rigorously done to figure out, what would be the best way to make those types of shifts happen. On the chemistry side, I think there's a lot of exciting ways where we can shift here. There's already a big push from the Department of Energy to essentially create enough green hydrogen within 10 years that essentially would allow almost all of Haber-Bosch to go over to essentially green ammonia production. So I think some of the basic chemistry and approaches there in the IRA Act are essentially already putting us on a trajectory where green Haber-Bosch is likely to be tenable. Second area is there's all sorts of ways we can make amino acids. Right now, amino acids are mostly being made through fermentation, except for methionine. But the catalytic production of other amino acids and precursors is an area being actively researched. And there's a range of studies that kind of suggest it could drop the price of amino acids by about 50%. So I think there's a lot of opportunities there where we can work with that. And then when we do have catalytic production of amino acids, we have to deal with the stereochemistry. And that's one of those areas where I think it's a combination of chemistry and what do we do on the in terms of microbiomes to deal with that stereochemistry. I think some of us have also been asking the question about what amino acids should be produced by which approach. And here I have a plot of essentially the nitrogen to carbon ratio of various amino acids versus their energetic cost. And what I'd like to highlight is essentially that there's about a 3-to-1 ratio in that nitrogen to carbon ratio, and almost a maximum 7-to-1 energetic cost ratio between various amino acids. And because plants are good at doing photosynthesis and fixing carbon, they're actually should be the things that are creating these amino acids that have high energetic cost. While the ones where we just need nitrogen attached to an amino acid, those are the ones where fermentation and catalysis can frequently play a really key role. And then there's some amino acids that kind of fall in between those two. So I think when we start looking at this and modeling across a larger system, we can start asking the question, which amino acids should be optimized on which types of systems? I think a lot of us have heard in the popular press, of course, about all sorts of alternative and cultured proteins. This is an area that's certainly going to expand over the coming decades. And a lot of it's going to be determined by how fast it expands and what it expands into by consumer preferences. But cost right now is one of the big issues here, and scaling fermentation systems for efficiency, I think, are really key. And then the flavor, nutritional biochemistry, and flavor perception, I think, are the other research areas that, in order to get consumer acceptance, are going to be really key to have this protein technologies move forward to a larger scale. On the manure side, and I'm sure Mark will go more into this, essentially there's a lot of opportunities in the area of manure sheds, of technologies to really transform and refine manure, the standardization of manure properties. And so once those manure properties are standardized, then it allows for more trading of manure across there. And essentially really an engagement of operators at the appropriate geographic scales to move those. So I think this whole combination of essentially shifting the amount of manure we're producing, the nitrogen compounds by providing more amino acids is going to shift manure, and then the standardization processing of it. I think there's a lot of opportunities here to cut down on greenhouse gases. One of the other areas that, if you look through all these different diagrams, you start realizing that microbiomes are key, whether that's in the soil, on the plant, in the livestock, in ourselves. The biology and the system modeling that is going on with each of these different nitrogen compounds is one of those areas where we, again, we're going to need research and discoveries to make the flow of nitrogen as efficient throughout our entire system here. And so let's now kind of get into the cropping and soil system side of things. As a maize geneticist, this is the area I've been most focused in on. And the area, what I would say, if you look across, whether it's a maize field or a soybean field, we've had tremendous success in breeding within a season to produce very high yields and high levels of productivity. But what I would say we failed in this is that these plants and systems cooperate within this growing season, but we've really failed to share and breed for the sharing of light, water, and nitrogen, phosphorus, and microbes between seasons. We don't breed for rotations or the interactions between one year's crop and the next year's crop the way we could with all the types of quantitative genetics and tools we have at our disposal now. And I think that's one of the key area where we need to shift on the plant breeding side and soil side, is how to breed for that system rather than just breeding for that 1 year's crop. And there's a number of different areas in this that we can do this. And I'll talk about a couple of these in terms of cold tolerance, but I also will highlight that there's all sorts of great research to be done in precision and novel nitrogen applications, rotations, and perennials in certain environments. So the project I want to talk about that's just getting launched right now here is a CIRCA project. It's about a circular economy that it's reimagining corn agriculture. And this is, I think, really a key one for the US, because in the case of the US, corn and the corn soy system is responsible for about 45% of the US ag greenhouse gas problem. And so it really is central to what we need to tackle. And so a group of over 24 labs across the country have come together to try to address this problem. And we're now just launching this project to work together. And so I'm going to tell you a little bit about our strategy and a little bit about some of the approaches we're using to go after this. And essentially what we want to do is shift that nitrogen soil cycle. So in our conventional cycle, these nice diagrams from Mike Castellano and models from him. We take a nitrogen fertilizer, we build up a large nitrate pool that the crop can tap into. But the problem is that nitrate pool is mobile. It can go into nitrous oxide, goes into leaching, and it creates a lot of those environmental problems and essentially inefficiencies in the system. And the one we're going after, essentially what we want to do is build up a much larger organic nitrogen pool with fast cycling between them. And so that it needs to be, and so that should result where the crop is able to get the nitrogen it needs, but then there's far less nitrous oxide and leaching out of the system. And then we are topping up the system with nitrogen fertilizer as needed. And a lot of this has to do also with getting the timing right. For when nitrogen is being used and available in the system. And so right now, maize nitrate uptake is essentially out of balance with when natural soil nitrate production is going on, which is mostly in the springtime, which is when we're getting a lot of this nitrate leaching and nitrous oxide release. And so the basic strategy is, how do we get a better match between these? How do we shift it so we can get the crop on the field at the time when that natural nitrate production is going on? And then how can we increase that soil organic nitrogen so that we have a bigger pool to pull from? And so essentially we have two strategies in the Circuit project. The first is nutrient recycling. And if we are building a system where our livestock are tapping into a larger amount of synthetic nitrogen, then we can reduce grain protein. We're aiming to take maize grain protein from 8% down to 4%, and then also reducing phosphorus levels by half. At the end of the growing season, after the plants and the leaves have used the nitrogen to fix everything, we are working to remobilize that nitrogen back down to the roots, just the way essentially perennial crops do, perennial species do at the end of the growing season. And essentially, the other area is to shift when nitrification happens. There's all sorts of compounds that plants exude and potentially residue products that are affecting the nitrification process, and the biological nitrification inhibition is an opportunity to shift that cycle around. And so we're going after some of the genetics that are involved there. And the second area is, we saw how important that spring time is. And that springtime is really important, of course, for nitrogen, but it's also one of the times when we have maximum light, useful light for doing photosynthesis. So what we'd like to do is be able to add as much cold tolerance into crops like maize so they can be planted a few weeks earlier, a month earlier, or even perhaps in the southern parts of the US, a winter annual, for example, in Texas or in the Carolinas. But essentially allowing you to get onto the field earlier and go out after, tap into more of that nitrogen and work there. I've highlighted in a couple of these that we're not showing rotation crops or other cover crops in the system, but I do want to highlight that we realize that in all these cases, it really is going to be a system, and that cover crops and just figuring out ways to get that land covered year round is going to be key in each of these different systems. So you put those two together, and what you're starting to see is we're going after a series of traits that essentially work together to get a better match with the natural nitrogen cycling and increasing the soil organic nitrogen. And so how do we intend to go after these various traits and add them to maize? Well, it turns out that maize has a lot of wild relatives that are already doing these types of things. They got a number of perennial relatives, and its sister genus Tripsicum is quite cold tolerant and frost tolerant. And so essentially, this group of scientists, we have the genetics available to us to essentially go sample across those. And then we also are looking at a group of about 600 species that are related to maize. Of these 1,200 species, we are analyzing about 600 of them. And again, this group of wild relatives of maize, the Andropogone, include everything from sugarcane, sorghum, miscanthus, and so on. They also are very good at nitrogen recycling. Many of them have readapted to cold and frost tolerance. And so as a group of scientists, we're working across these different systems to tap into these traits. And so, we realize that, just not one of these approaches is going to be the only solution, but they really need to work together with some of these other ones. And so, and I'll show you a couple other points here that we think are really key. So I think we all agree in this, those who've looked at it, the land needs to be covered year round with various strategies, and that could be cover crops, perennials on some acres, and of course trees are the other ones. I think there's a big opportunity to essentially start doing multi-cropping and with an increased usage of things like winter legumes. Winter pea looks to be a really exciting opportunity where we could combine that with corn in a much more efficient system. We don't do a lot of this in the US, but globally, there's a lot of burning, residue burning that goes on. And essentially, just that alone is producing about 3.5% of global greenhouse gas emissions. And we're not talking about the CO2, we're just talking about the methane and nitrous oxide coming off of those. That remobilization of nitrogen back down to the roots would substantially address those nitrous oxide releases coming off of these burned fields. And then I think there's a lot of key, again, in rice, obviously not a giant crop here in the US, but globally, rice, there's some key areas here we can also address it. And there it has to do with essentially modulating water, dry and wet periods, again, to deal with the soil and nitrogen plant interface. And so I'm going to end there. Mark's going to tell you a lot more about the whole livestock and dairy side of things. And I'd be happy to take some questions at this point. I think, is that how we kind of wanted to do this, Jack?
Yes, that's correct. So yeah, thanks very much, Ed. Very fascinating, very thought-provoking. Lots of great ideas. And we got a number of questions in the chat. Some of them have already been answered by other members of the audience. Let's see. First question. Early on you had a statistic of that agriculture contributes 40% of U.S. emissions in the future on a yearly basis. Is that on a yearly basis, or is that cumulative since some time period?
No, that's the year. That would be by mid-century. That would be the yearly emissions from agriculture. So right now, agriculture is at around 11% of U.S. greenhouse gas emissions. But the when you start electrifying and doing a lot of these other things, the denominator gets a lot smaller. And so agriculture, essentially doing, what we're doing right now, which is we are absolutely getting more efficient. I don't want to, highlight that, a lot of the gains are because agriculture has already gotten a lot more efficient. Globally, agriculture is probably responsible for anywhere from about 20% of global, 20 to 25% of global greenhouse gas emissions. And a lot of that is from things like inefficient livestock. Mm-hmm. And so a lot of the successes we've had, the reason we aren't at 25% is because of what we've already been able to accomplish and everything. And so I think, so that's, globally, we need to apply some of the learnings we've done here elsewhere.
Yeah, and I think that's often forgotten in a lot of these discussions, that I think the most effective way to reduce these inefficiencies is to improve yield, and that's both on the plant side and the animal side, that improving yield reduces or improves sustainability. But like you point out, we can do a lot more, and
right.
we need.
And yeah, and that's where like winter pea, that essentially now you're double multi-cropping. That's increasing productivity per unit land. And so virtually, if we're doing it right, productivity goals should be totally in line with this. And putting nitrogen in a grain is one of the most expensive thing a plant does. Okay, so by just having plants make starch, that's a lot easier for them to do than packing them in with protein. So we would get productivity gains there, while we're letting Haber-Bosch, which is very good from efficiency, to do what it's good at doing.
Yeah. So at one point you talked about that we really lack modeling in the area of modeling nitrogen flows. One question is, well, what data do we need to enable that modeling?
Yeah, so I think we need a lot more data that's essentially a cross-scaling, multi-scaling model. So where we have data at that plot level, farm level, up to essentially the regional scaling. And there's been some excellent we kind of know our total nitrous oxide budget and numbers from that fairly well. And then there's been some great studies that are showing with very good temporal studies, in certain fields and and things that are telling us these things. But we really need to, given the nitrous oxide and some of these other things, you get pulses of them, and they can be very sharp pulses at very short periods of time. And so you would really like to know over millions of acres what's going on over a couple-week period, okay? And be able to model that accurately, so we know what's the right type of system to stick in each one of these systems. So I think we understand the basic principles now of what the problem is, but there's going to be the solution is going to be is certainly very regional.
Yeah. There's a question about limitingness of amino acids. I'm going to leave that until after Mark's talk. But then there's another question about let's see. Oh yeah, what do we have the data now to breed? I think crops that are have higher nitrogen efficiency, or do we need to collect additional data?
Right. So one of the things I would kind of highlight is that I think we've already been doing a really good job breeding for what we would normally call straight nitrogen use efficiency. So you take down whatever the yield is, per nitrogen, or the amount of nitrogen in the grain relative to fertilizer put in. I think the issue is that that was efficient for the crop. That wasn't system efficient. That's the key thing, is it would have been more efficient for having a plant make the amino acids is not efficient relative to doing it synthetically. Okay. So that's the basic problem is breeders have been selecting for nitrogen use efficiency, or nitrogen in the grain relative to nitrogen applied sometimes. And it would have been more efficient for certain purposes to just make the amino acids, or give that directly to whatever needs it.
Yeah. Okay. So a related question that I have is, you talk about breeding for a shift in cold tolerance, and you talk about looking at genetics in other species or related species. So what about the genetic variability within the pool of elite germplasm? Do we have the data now to be able to select, and is there genetic variation to already start selecting in that direction?
there's some. So we know, for example, highland varieties of land races. I would say in the elite varieties, doesn't look like there's a ton. U.S. elite varieties doesn't look like there's a ton of variation for additional cold tolerance. Europeans have definitely been selecting on it in their environments, and there's, I think, some opportunities there. And then the land races of maize, these very highland Mexican, highland Indian materials certainly have a lot more cold tolerance than is out there. And then, you know, we've already started screening various introgressions and things like that, and can see that there appears to be substantial variation out there. So, and then these wild relatives, you know, some of these are very close wild relatives. And you know, some of them, you know, can get down to -7, -10 degrees C without damage.
Okay, so final question before we go to Mark from the chat. On the topic of introducing perennial traits, do you have an opinion on how we should integrate? The John Innes Center recently published on the AG2PI allele for cold coping. Would gene editing be the desired tool?
Yeah, so for some of these traits, you know, that's why we, in our that circuit project, we're doing both introgression and you know, and where we can't, where this, you know, actually, even if it's in Triptychum dactyloides, there would be ways, if you wanted to, we can make a cross between those, and we could bring those alleles across even between the sister genera. So depending on how hard you want it, you could do it without transgenes or editing. But yes, we're going to test out hypotheses with lots of editing and transgenes. How industry decides to finally deploy it would depend on, you know, regulation and market conditions at the time.
Okay, good. Well, I'm going to leave it here for now, but yeah, we'll bring you back for the second round of questions. So don't go anywhere. So turn it over to Mark. Mark, please go ahead, share your screen.
I'll do that. Thanks. Thanks, Jack. Are you seeing it?
Not yet.
Okay, hold on. One more punch. There we go.
Here it comes.
Got it?
It's still waiting. It says that you've started screen sharing.
Oh my, okay.
See it yet? Maybe just slow internet, I don't know. Yeah, here it is. Go ahead. Yeah, we see it.
Yeah, sorry about that. We are not best internet service in the world here in Nebraska, so apologize. So I was counting on the talk that Ed just gave, right? They have worked on this concept, the circular approach to these, looking at these economies for circuit and all kinds of things. And that's just fantastic. And they're way down the road. I'm going to touch on what we're doing with the CENSA effort with respect to animals and manure and microbial systems. But the point I'm going to really more focus on today is how far behind we are, right? When we think about optimizing or improving or looking at nitrogen use efficiency through the livestock systems, how much of a challenge that is, how much more complexity there and expense there is, and really touch on some things that we're trying to do, have been working on for several years, and we're trying to build out with respect to frameworks and looking at how we are approaching some of these challenges, and really just kind of harp on exactly what some of those are and how we're looking at it. And then realizing what a value this concept that Ed's presented brings to us on the livestock side with respect to potential interactions with expertise and building on successes of others and really finding opportunity for funding and all kinds of other things. So Ed showed you this, and he talked at great length about the diagram, and I hope we're keeping up. Are we, Jack? I have the diagram up now. And how we developed that, and that was excellent, right? And so really appreciate those. So again, a little change of pace. So if we think about the center approach, there's two major components that we would say focus in on livestock, right? And those are our livestock nutrition systems talked a lot about amino acids, and in poultry and swine, we do use a lot of amino acids to limit the amount of crude protein that we feed to those animals. We base that on the most limiting amino acids at the moment, right? We have done that for a generation. And in swine, for example, that's lysine in most cases. In other cases, you can get methionine or other limiting amino acids depending on your feed ingredients, but more focus on that. And now we're doing a lot of work here at US Mark on amino acid supplementation to feedlot cattle instead of crude protein. So Ed's on to something. We're dovetailing there with the work that we've been doing in the past, and now what's the opportunity to expand that? So the price point is important, right? Lysine is essentially marketed based on the value of crude protein, and how do we now make that more advantageous to feed with respect to those animal systems and the nutritional requirements and the multiple rations that go into a poultry or a pig production system, and how is that all managed? And then the whole area of microbiomes related in the gut and the rumen is just in its infancy, and just a lot of work to do there to basically understand how those systems have evolved and how they're anchored and how they move and provide value to the animal, let alone the management and improvement of those systems. There's a lot of work to do there. And then manure management comes and goes, right? In swine and poultry, it's relatively effective. We're doing a better job all the time. I still got a lot of problems with too much or too little, as Ed pointed out. In parts of the country, manure is just piled. It's just not really very valuable. They don't have the leaching issues. They don't have the opportunity to capture that nitrogen. And we just have to do a lot better. We're talking about things like how do we limit ammonia losses on feedlot surfaces, all kinds of things. So again, we know a lot about what we're doing. We're putting a lot of energy into those concepts, but there's a long ways to go with respect to making these systems much more efficient and much better from an optimization perspective. So if we talk just a minute about the initial development, so Ed challenged us to come up with some ideas and some teams. So more or less because of the work that I'm going to present a bit of today on the beef side, we're kind of providing some leadership there. In dairy, these are ARS leads. We have Dennis Hancock in the Dairy Forage Research Center. Swine and poultry, the ARS lead is Brian Kerr in Ames. Manure management is Pete Kleiman and others in Fort Collins. And microbial systems is a diverse group, but some leadership from here and some leadership from the Dairy Forage Research Center. And we've pulled these teams together and they've been organized, and some of you on this call may be part of these because they do include rather extensive university partners. Again, these are just kind of an executive level teams because we're waiting to see how the program gets rolled out from Ed's Circa program, where the funding and opportunity might lie. And this is relatively new to us. So we're more in the think tank stage here, developing the concepts around how we would move these programs forward, feeding off of Ed's leadership, which has been extraordinary, and the group that he's put together already that's done the great work that he just presented. So we're more or less in our infancy, but we do have teams put together. We do have great leadership. We do have great representation. Each of these groups has developed a white paper, and that's to inform the others and to provide some staging opportunity to look at how we're going to evolve in and out of ARS to develop this whole CENSA concept more completely as we move down the road, right? So again, the primary areas of focus, I'm really not going to touch a whole lot more on this today because of time, but feed efficiency, right? And the nitrogen use efficiency within that framework. And your comment, Jack, about just making things more efficient, right? That's important. Protein quality, amino acid supplementation, meeting those nutritional requirements in a much more fine-tuned way, and creating higher quality protein products, milk and meat, think about off the other end of the system, right? Touched a minute already on rumen and gut microbiome, and I'm not going to dive into the science there at all today, but tremendous opportunity as I see it, but a long, long, long ways to go. And then all kinds of genetic and genomic implications around just selection for efficiency, selection for adaptation, and other things which I will talk more about. And then, of course, how do those management systems evolve and incorporate and Field peas, for example, is something I need to think about here in our big system where we have 34,000 acres and 7,000 acres under pivot irrigation and 20,000 head of livestock. And how do we start to be a better representation, provide better representation of industry-based production systems, and how do we incorporate all this is always top of mind here for me as an administrator, right? So we talk about some specific opportunities for livestock. Well, genetic and genomic technologies, again, if you think genome to phenome, we are revolutionizing a lot of these technologies. In an era of genome editing, there's just unlimited opportunity. It's finding those low-hanging fruit and using those to drive improvements and to look at this on a global scale, right? Feed efficiency and nitrogen utilization and improving productivity and production efficiencies on a global scale. Much better optimized feed and forage use efficiencies. For example, reducing nitrogen inputs, just exactly as Ed talked about, increased use of synthetic nitrogen, et cetera. And then more precision and increased feeding system and protein quality. And with the goal, hopefully, to provide a reduction in feed per unit of production by 30 or 40%. I don't know, those of you that are familiar with animal production, that sounds like a big number, but even in relatively uniform genetics here at US Mark, we see a twofold difference between the intake of one cow versus another in the same genetic line over a year, right? Her maintenance needs, those maintenance needs are extraordinarily variable. And we see, you know, feedlot efficiencies of 8 pounds of feed, for example, per pound of gain down to, I saw a bull come off test this spring in Iowa at less than 3, right? So there's just tremendous variation and opportunity to look at this feed and forage system combined with genetics to really provide a lot of improvement. We're guessing actually about optimizing the ruminant gut, right? We think there's potential there to be huge opportunity to improve health and feed efficiencies per unit of product produced, maybe 20 to 30%. And at the same time, reduce total methane emissions by 80%. And there's a lot of intentional work around methane going on. I'm going to touch a bit on that today, but more all the time and a very high priority for this administration, right? So there's just a lot of gray area there, but maybe a tremendous opportunity that's yet to be realized. A few more to touch on: integrated production systems that maximize production efficiencies, right? Not production, but production efficiencies, nitrogen use efficiencies, and improve animal protein quality. So think about soil ecology, the things that Ed was talking about, right? Cropping systems, manure management. How do these nitrogen and carbon cycles affect all that? Biodiversity, ecosystem services on rangelands, et cetera. And then we see it all the time, right? Especially here where we're at in Nebraska, our weather extremes, extreme weather events are going to dictate more and more how we select genetics, how we select cropping systems. You know, a beautiful cornfield that's been planted in March and looks great and gets hailed out in June is not a lot of value, right? And that's happening more and more often. So how do we adjust for those and add this level of complexity into this question is yet to be determined, of course, actually, right? And we've set some goals as along with industry. So the beef industry, National Beef Cattlemen's Council, NCBA, sorry, their goal is to be carbon neutral by 2040. U.S. Dairy has a goal to be carbon neutral by 2050. Goal to reduce methane and nitrogen-related emissions by 80%. The pork industry would like to cut greenhouse gas emissions 40% by 2030, right? And then we have all kinds of other goals around different aspects, different stakeholder groups, et cetera, all being brought to bear and being considered as pretty audacious goals and things we need to strive for. So setting those milestones, setting those goals will be important as we look at developing these programs, right? So I'm going to spin out of that CENSA focused area just a little bit and circle back, okay? So we have been developing a beef research consortium approach to our cattle programs for several years. I'm going to touch on what's the holy grail for beef production. And I'm going to speak mostly about beef production here for the remainder of the talk, only because that's where the big methane opportunity is. that's closest to our heart with respect to how we're looking at these systems and trying to improve those. So you all know the problem and the challenge, right? Climate change, food security, climate adaptation. Yet there's 13.5 million square miles of rangelands on Earth that aren't going to grow lettuce anytime soon, right? So there's an extraordinary opportunity, and there's all kinds of extraordinary opportunities on the horizon. Some that Ed touched on, there's lots of others, things, genetics and graphene and all kinds of things, right? So we know there's opportunity. We know that there's going to be tremendous change. How do we position ourselves to be on the forefront of leading those discussions and to integrate and demonstrate those opportunities? Also, a whole lot of focus now, right, on systems, and that's what Ed's talking about. It has been for years. Animal systems are catching up. It's a very high priority in the national academies, et cetera. So you all get that, I think. And then just embracing complexity from an animal production system relative to nitrogen or anything else, right? Where we need to develop the mindset to be able to program around these challenges. So we call it GEMS. It's genetic by environment by management system. And we've added S recently in the last few years, which is the socioeconomic factors that you all are more or less aware of, mostly aware of, right? That are starting to dictate how we look at these complex production systems. But the systems approaches are very difficult to describe, justify, and particularly to fund. I'm going to touch on that a bit. And every time we look at it, right, Jack, it becomes more complex. And we just have so much that we're weaving together. And how do we now pull all that down into bite-sized pieces so that we can dive into things like nitrogen use efficiency, right? So the Holy Grail is what? Well, the Holy Grail, as far as I'm concerned, is perfect optimization of a beet production system on a landscape. So we know what the resource is, we know what the value of that resource is, we're ensuring and protecting that resource, and we're producing a high quality product on the other end in the most efficient way possible with the tools and the resources that we have, including nitrogen. And so instead of saying we need maximum yield or maximum production of that beet production system in the future, we need perfect optimization or maximum use, efficiency use of that resource. So that's the spin, right, that we're really trying to promote. It's difficult, right? So many of you are probably not part of the beef industry, so I put this slide in here. And clearly you can see a lot of variation there. I think Jack could tell you what all those breeds are, but they all do different things, right? They have different roles. They're not the same. They don't have the same physiology, growth, health, efficiency. They have different product yields. Holstein bull in the upper right is mainly for milk, right? They have different heat and cold stress adaptations abilities. They have different resistances to pests and pathogens. They're adapted different environmentally, et cetera, right? And you manage and you develop these animals completely differently. But the same can be said for similar genetics. Can't really say identical, but similar genetics in very diverse environments, right? So you have Angus cows, for example, in Montana versus Angus cows in Alabama, or Angus cows in Argentina, or Angus cows in Eastern Europe, or wherever, right? Just because of the management system, the G by E by M, just because of the differing E and M, right? You have dramatically different cattle with respect to how they are adapted to weather, the forage base and the biodiversity that they're exposed to, how they're managed, predator, pests and pathogens, et cetera, right? They are different cattle and they have to be managed differently, and we need different tools. And we need different systems that optimize their production and their resource use for each of these herds of cattle in very, very different ways. And I think that's maybe the lesson that I'm trying to really harp on with our leadership, is that the one size fits all approach to nutrition and management and health and genetics just doesn't fit, right? It doesn't fit at all. And we have to start really focusing on tools, robust models that now give us the opportunity to optimize the performance of these two herds of cattle. Even though the genetics are set the same, they have completely different, not completely, but they have very unique production system requirements, and they need unique management systems. So going from there, we've been working with University of Nebraska at Lincoln and others on a beef systems research consortium And again, we're not going to dwell on this very long at all, but I wanted to give you a quick overview of it. It's really a framework and an organizational structure. We're not really talking about projects like we are in CENSA, where we're going to start to drill down. But hopefully here in a minute, you'll see how this fits together. So we've been working on this for a while. And these are slides that I use for stakeholders, but I'm going to jump out of them pretty quickly and just show that when you're talking about a system like this, right? A production system for beef cattle. If you want to call it a sustainable beef production system, it starts and ends with soils, soil health, rangeland ecology, rangeland health, that kind of thing. But it learns and interacts with the forage systems, right? So we're focusing on better soils and better pastures and better forages. The forage system interacts with the cow, kind of to the point I made on the last slide, right? How do you learn from year to year from each of those systems, pieces of those systems, and how do you improve those? How do you learn and iterate those? How does the beef cow interact with soils through manure, grazing patterns, drought, water quality, all those things, but manure management and nitrogen and phosphorus. And of course, the end game always has to be that we're supporting high quality protein products into the meat case, right? And I'm not disagreeing at all with Ed that we'll be seeing cultured products and alternative products in the future, but there's just too much of unimproved rangelands around the world that need managed for carbon, need to be managed for nitrogen, and that best use of those is always going to be some kind of a grazing animal, right? So I think these systems are going to change dramatically, but I think the role of the rumen and the use of lower quality forages that monogastrics and humans are not able to digest is going to remain a very large part of the equation going forward. So our challenge is to optimize these systems, to maximize the efficiency, and to begin to think about how we pull the nitrogen out according to the things that you saw from there. So this looks relatively simple when you throw it all together. Even this, though, gets to be crazy, and this is just a flyover slide, but on the left are all the different GEMS considerations at each of those junctures. There's global objectives, there's all kinds of partners in blue, and it's just, it becomes very daunting, right? So how do we get our arms around optimizing these systems and looking for opportunities and low-hanging fruit and long-range objectives relative to science? That's the challenge. So we're trying to understand these big overarching objectives, right? We want to optimize or focus on environmental sustainability, nitrogen included, production capacity. We need more efficiency. We need more economic sustainability to go with these systems around the world. We need high quality beef products. We need the safety pieces to go with that. We need to focus on animal health and husbandry, care and well-being, right? We need to weave all this together, and we need a lot of support on communication, marketing, and outreach, extension, et cetera. So those are original overarching objectives. So let's drill down just a bit. So we think about how we're trying to organize this and how it fits with CENSA. Well, we have developed what we call modules, and we're putting together teams now that focus on these. So there's 10 of those. There's soil, water, and ecosystem services. There's pasture and rangeland forage systems. There's genetic genomics and animal breeding, nutritional physiology and lifetime productivity, health and well-being, product quality, safety, healthiness, and nutritional value, precision ag and on-ranch implementation of related technologies, complex system modeling, things you heard from Ed, production system, economic viability and resiliency. And that's the first thing the cattlemen tell you that they'll need, right, before anything else. And then diverse socioeconomic factors and implications for future beef production and beef production systems. So we're just getting started here, right? Trying to develop these teams, and we're working with UNM and other academic partners, including Iowa State and others, at least with some initial conversations, but more closely now with Colorado State, South Dakota State. We're looking at how do we coordinate resources and eliminate redundancy? How do we describe these complex, cross-cutting, strategically valuable challenges with stakeholder guidance in the way that Ed has already done so with CERCA, right? What's the next step for us? And not only in beef, but in dairy and in poultry and pork. And what are the gaps? How do we get to those? Who should be on these teams? And how do we absolutely make sure that we're not reinventing anything, that we're building these bigger networks of collaborations I'm speaking primarily within ARS, but with collaborators as well, right? And diverse partners. So we do have a lot of partners already in place. We have ARS scientists across a dozen or so beef production labs. We have University of Nebraska, Colorado State, South Dakota State, and others, including hopefully Iowa State at some point. We have NCBA and state cattlemen groups working with us. We have other federal partners, BLM, NRCS, and others that are potentially going to help inform parts of these discussions and be part of our advisory groups, if not part of the research. Looking at others, right? Research and industry partners internationally and other stakeholders across the beef production and livestock production spectrum. So where are we at on the process? We have a great plan. We have a white paper. We have been working with these other groups and starting the meetings. We have Colorado State, South Dakota State, and Nebraska in next week, right? To talk about how we can start to look at the systems-wide look and leverage resources. So we're having good traction, really, very good traction, but less so with leadership and USDA and maybe with funding opportunities. So the opportunity with CENSA is paramount, right? How do we catch up to the point where Ed has taken his programs? How do we better position the work in livestock? And we have pork programs here at US Mark along with sheep and cattle. And how do we look for funding to develop these things out and push this forward and working together and relying on Ed? We do have a little new funding this year for the first time in a while for greenhouse gas monitoring. I think that's interesting that the label is monitoring. I'm not sure where we're going to be taking that, but one thing we are going to try to build out is a very highly accurate measure of lifetime productivity of methane in extensive cattle systems. So how much methane does that cow produce based on her GEMS orientation, right? Her genetics, the environment she's in, how we're managing her. And then hoping to build on success. And we have extraordinary success across ARS in genetics and nutrition, et cetera, in all species, and with, of course, across the land grants that we're partnering with. So there's opportunity. It's just how do we get it positioned? How do we leverage opportunities to really well support CENSA and the great objectives that are laid out there with respect to nitrogen and greenhouse gases in general? And what are our opportunities here specifically? Those are the questions we're trying to address right now. So there's lots of work left to do. It's very exciting to see this move forward and really appreciate Ed's leadership and the opportunity to participate. And we're looking for partners and ideas. And I'm happy to talk to any and all of you after this meeting if you've got opportunity ideas and you've got things you'd like to share. So just a couple quick examples of how we're trying to move this forward. We have huge commercial populations here, 8,000 cows, the biggest research herd in the world, actually, that's on one site. And so we want these herds to be representative industry populations, and we want to really focus now on developing, number two, optimal production systems, right? So modeling effective practices. I want indexes for maternal, terminal heterosis, all back to Jack's point about improving production and production efficiencies. We really want to focus on demonstrating selection strategies that are robust and help us optimize these systems across diverse environments. And we really want to show the compounding effect year over year of optimizing these systems, improving genetics. And that's the microbiome, that's climate resiliency, that's genome editing. There's just a lot going on there. So the most famous herd of cattle we have here is called our germplasm evaluation project. This project was started in 1972, and it currently is evaluating 18 different breeds. And there's nothing to learn here, just to say that this is a really complex organizational challenge for the people doing the breeding and the AI work and the management. But what we're learning is how do these different breeds look one to the other with respect to growth and efficiency, et cetera, health, and how do those crossbreds interact with varying levels of heterosis, et cetera, in one environment. That's the challenge. We've been doing it here, and it's been very informative, and it's really promoted a lot of value to breeding systems across the world for beef genetics, but it's here. It's not in maybe different environments, southeast, north, south, whatever. So we are looking to expand potentially to additional breeds. We're looking to evaluate some of the large composite programs. Some of the biggest herds in North America don't use purebred bulls, for example. They have their own genetic lines. And we're going to add now with the methane work and with the ties to CENSA and some new money, right? How do we start to evaluate these current breeds for novel traits, particularly things like nitrogen and greenhouse gas emissions? We have expanded this herd into Texas and Oklahoma just in the last couple of months so that we can start to look at G by E across much more diverse production systems, and particularly with relationship to heat and heat stress. The cattle in Texas are clear down by Corpus Christi, for example. So tremendous opportunity there. It fits with CENSA. It fits with the focus on production efficiencies and how do we look at nitrogen use efficiency. And again, we're adding some funding and some new metrics and monitoring opportunity in support of CENSA with some new money from ARS, right? So just touching on that as an example. So what's it mean for us? Well, we're evolving our bee programs, right? More focus on sustainability, nitrogen and methane. We're looking definitely for larger collaborations and much more diverse G by E by M analyses, right? These are some of the folks that we're talking to. We're looking for funding as always. We do have a little new funding, but I think we've got a lot of great ideas. And we really, again, I'm just going to circle back to the technical expertise and the leadership that Ed has brought to this discussion really anchors everything that we're trying to get to now on the animal side. And we have a long way to go. But I do think we have a good start, and we have pieces in place that are going to start to inform that. So not sure how much time that took, but I didn't want to go over, Jack. But that's what I've got today to share with you. And happy to circle back with more technical information as appropriate, for example, with beef or swine poultry or anything else or dairy. I didn't think I had a lot of time to dive in there.
Okay, thank you. Thank you very much, Mark. Yeah, and you're right. I mean, there's huge complexity, and I think I also agree that one of the ways to tackle that or get a handle on that complexity is modeling, and Ed talked about it also. If you can develop the model, and they don't have to be perfect, you can build on them, but that allows you to formulate a lot of the questions or identify where the gaps are. So yeah, again, put your questions in the chat. I want to start with a question for probably. Well, actually, part of this is a comment. So Mark, when you talk about funding, looking for funding, I think AG2PI is a source, at least for funding related to genetics and genomics. And just want to remind everybody that NIFA a couple of weeks ago sent out a new RFA for AG2PI, and I think the proposals are due. June 1, still not the 40 million that Congress has talked about, but I think a little over 2 million. And different from previous grants, there will be several research grants that will be funded directly through NIFA, so not through the way the current AG2PI has operated, that it comes to in this case, Iowa State, and then Iowa State provides seed grants to a lot of the scientists and industry, but it goes directly to NIFA. But hopefully, in next year, that program gets expanded to a much larger dollar amount. So I think there's opportunities now to apply for funding for some research projects in this area and other areas, but hopefully more in the future. And then I also want to point out that we will have, as part of the current AG2PI project, we're organizing a conference in Kansas City in June, and where we will also identify or have discussions to identify gaps and formulate approaches to tackle a lot of the questions that were raised here also. So hopefully many of you can participate in that, and registration is out there. If you go to ag2pi.org, you can see a lot of the information, but hopefully we can continue a lot of these discussions there also. Mark and Ed, with regard to members of the audience that want to contribute, that want to participate, what's on the crop side, on the animal side, what's the best thing for them to do? And it's both industry and scientists, academia.
Go ahead, Ed.
Yeah, so if somebody's interested in the corn project and stuff like that we're working on, just feel free to reach out to me and we can kind of hook you up with what we're kind of doing and everything. We're just getting started right now, and already a number of folks who are just interested in the project, even though they weren't necessarily directly funded, are already saying, hey, let me work on this area, and we're working together on it. So I think there's opportunities there. On the larger CENSA effort, I kind of highlighted some of the key people in those different areas. Mark on livestock, Peter Kleinman on manure, Lisa Ainsworth and Virginia Jin. And so I think I would maybe reach out to each of those folks to kind of see where we are in those different areas. I think, as a USDA, as an effort, a lot of this so far has been percolating up and within ARS. And I think in the next, few months to year, I think it really needs to have a lot more collaboration with the rest of the folks in REE. There's a lot of excellent questions being raised here on the economic side. Those are really areas where ERS and on the NIFA side are really the specialist on. And I think we are at a stage now where we need all these different elements starting to come together. And and we need to have funding structures that really kind of pull together, that bring together the partners on the two sides. And so far with Circa, we've been using FR and ARS have been kind of essentially funding the two elements together. So that's how we're putting it together. But I think longer term, it needs to be a partnership across REE that supports this.
Okay.
And that's where Ed's real value is. And I think that's exactly right, Jack. If you're interested in the livestock pieces or anything that I shared today or anything that Ed shared that would be more relevant to livestock, absolutely contact me. As you know, though, if you're in livestock, that the funding is daunting, and we definitely have some low-hanging fruit, right? But it's behind pretty tight fences. And so, for example, we're developing a technology with a simple little single edit. We produced a calf that's completely 98% or so resistant to BRD, right? Bovine respiratory disease. But to get that calf into a position, or that technology into a position so that it can be commercialized, when we think about editing in livestock, that's roughly $500,000. And that's for one very simple edit. And that's the challenge. We got dozens or thousands of opportunities to improve the efficiency and to look at these things, but the funding is just really extraordinary and a big challenge. And then, as you could see from the things I presented, you just get so many people involved in these complex systems that it's hard to know where to start. So we need leadership. We need everybody that wants to participate, absolutely. And then we'll start to build this out a little better. And to Ed's point, it needs to come together with our collaborators, if nothing else, relatively soon across REE. But you're all welcome, and we're happy to talk about it. And we're trying to find the time and just a little money to get us into these discussions at the moment.
Okay, good, thanks. Circling back to a question from before the switch of speakers. So for synthetic amino acids, is the relative limitingness of amino acids for livestock production, how should that be considered? And you talked about, some crop systems are better suited to produce some certain amino acids. So how should the limitingness of amino acids be considered when you look at the system?
Right. Yeah, I mean, I think, so I think limitedness is absolutely a key thing, and that's what we've been focused on up till now. But I think there's opportunities, for example, to start producing large amounts of things like leucine and isoleucine that are certainly not normally limiting, but essentially just are at the heart of replacing amino acids in the system. And so absolutely, you first focus on whatever's limiting, that's number one. But if you look at the price of amino acids and you start incorporating digestibility in there, and when I've talked to people who've actually been in amino acid production, right now most prices of amino acids essentially are just shadowing whatever soybean prices are. The real cost is still substantially less. And I think that's where this economics, what should be being produced, do we need more production here in the US? Is this an opportunity for where our ethanol plants, if they shift to new markets, those could be resources that could be deployed for producing, for example, like amino acids. That I think there's a lot of opportunities there. And when you talk to people about catalytic costs, so right now amino acid lysine is $1.50 a kilogram. I understand from some folks the cost is only about 80 cents a kilogram, and folks working on catalytic amino acid production think long term they could probably bring it down to 40 cents a kilogram. And so I think those are the, in terms of cost efficiency, that's kind of where we are at in these systems. And so absolutely limiting this, you've got to figure out what your feed is, and but I think synthetic chemistry can do a lot.
Okay. There's several questions that Sandra had, and I wonder, Sandra, whether you could unmute and just ask those questions directly. Some of them deal with EPA models that I'm not familiar with.
Yeah, sorry, Jack. Yeah, thank you for both of your talks. I think I was just wondering, what is being considered in your system boundaries? So it's clear that you are thinking, beyond a reductionist viewpoint of just yield for an annual season. But beyond that, we have more temporal consequences like climate change, and then also spatial consequences like equity in other nation states globally. And maybe we don't even have to talk about a global scale. We can just talk about state specific, like within the United States. So how is CENSA or CERCA Ed addressing these issues?
Yeah, well, in the CERCA project, a lot of what those the boundaries are, I would say the first thing is we're just starting out. Okay, I mean we're literally submitting the revisions of the grants for the final budget right now. But we have a team of about 6 modelers in there, and they are modeling. There's folks who are kind of Bruno Basso is modeling at a certain scale, and Charlie Messina is modeling at that plant field scale. And so we have people kind of working across, and so the modeling will be at those different boundaries, of what's the farmer impact. And then I've certainly been talking with groups in economics about the modeling that needs to go on there at a nation level or international scale. And and I think they're areas that are open. We I think we need to figure that out and model and ask these questions in a robust way. And I think the main thing that I've sometimes seen in some of these modeling approaches is that we don't have enough integration of people who know the technology or the physiology or something about this biological system we're working with the modelers. And I think that's the key thing that's frequently missing, is we need to have both parties sitting at the table And, because you may be able to say, oh, well, we can do this amino acid thing. That's not, that's something that we can shift. So I think there's tons of opportunities in modeling, and where that boundary is of course very important, but we need to test and evaluate these at different boundary points.
I have a question for Mark. Mark, you didn't talk about insects as a way to process manure or aquaculture. How do you see opportunities for integrating insect production or use of insects and aquaculture within these integrated systems?
So, Jack, that's a great question, and the answer is absolutely, right? And maybe there's more opportunity there than even we realize, but we have not really picked that up, right? We've been trying to get our other ducks in a row where we do have critical mass and expertise, and there's low-hanging fruit, right? So, for example, we keep talking about ammonia emissions from dairies and feedlots. How do we bind that? How do we not let that get away from us? Maybe that's insect related, right? I mean, we've got to start looking at really ingenious opportunities to take out the variables that we know are big contributors that are crystal clear to us, we just can't get to, right? In aquaculture, we definitely need to consider that. We have programs in ARS. We haven't done it yet. Our focus has been much more on methane to this point, but that one's been nagging at me, right? Why don't we include that? And that's, we do have a lot of great people, a lot of work, even in ARS going on there. So there isn't any reason not to consider them. We just haven't added them at the moment. Again, great point, great question. Something we need to think about.
Yeah. Another question that actually Sandra raised earlier, but I want to add on to is breeding systems have adopted since the Green Revolution, mainly for high input and high management crops. So she's saying that, well, maybe we need to breed in limited nitrogen environments to really increase nitrogen use efficiency. But I want to add to that and say, well, how do you incentivize breeding of crops that have lower protein levels? So how do you do it, and what incentives do you need to put in place?
Yeah, I mean, yeah, there's the sticks and the carrots. Okay. Obviously the stick-based approach is you start taxing fertilizer. Okay, but I would, that's, whether or not that's the right way to go about it. I mean, I think the IRA has been really interesting in hitting mostly a carrot-based approach and being able to get a lot of economic movement very quickly. And so if you go to the carrot side, that would be the type of thing of you pay at the grain elevator for bringing in low protein corn. You give a bonus. We used to give a bonus for high protein. You give a bonus for low protein, because then that means you left the nitrogen on the field. And so that would be one strategy. And then I think there's all sorts of opportunities for most, right now we import a lot of our fertilizer and we import tons of our amino acids. Every one of those things, the US would be superb at making in a renewable fashion. And we should.
Yeah.
And yeah.
The drivers are always, and Jack, I mean, are always regulatory or economic, right? And so there's a tipping point there, based on economic value, and there's no reason that lysine costs $1.50 to Ed's point when it's only 80 cents, but they back solve that according to what soybean prices are, and they market accordingly, right? So how do you negotiate those landmines? All we can do as a scientific agency is put the technologies together and push those ideas forward. And we hope that, the bigger brains will figure that out. And that's part of the challenge.
Yeah, because that same question also is, present in livestock now. There's no value to a breeder, to a producer, to a farmer, rancher, of having cows that produce less methane.
Right.
So, yeah.
None, yep. Maybe
yeah.
just the opposite, maybe restricting methane restricts the functional value of the rumen, right? And then you got a problem. So there's a lot of landmines there, and I like Ed's stick versus carrot line, because that's what it's going to come down to.
Yeah. So question in the chat. Yeah, as far as introducing a trait into cattle by gene editing, 500,000 you had mentioned, Mark. Question is, well, does that include the cost of getting it through the regulatory approval system?
So this isn't even introducing a gene. This is just modifying one, right? This is just stripping the ability of a pathogen to bind. I think I said BRD, it's actually BVD, viral diarrhea. So no, see, that's the problem. It's expensive just to get enough livestock on the ground to demonstrate that the gene works and that it doesn't mess up anything else, right? And it doesn't create any catastrophes with respect to consumer acceptability, those kinds of things. Then you got to get it approved. So it's daunting, and we do it. You know, we do a lot of genome editing at the junior college level, high school level now in plants, right? But not in animals. And it's not any more difficult. It's just much more sensitive and much more expensive. And that's part of the challenge, right? So there's opportunity, right? But yeah. So the point was the GE salmon. I'm looking at that comment. Was 50 million, and that's not even in new money, right? That's in old money. So yeah, I don't know. That process has to change, right? I think you'd agree, Jack. That process of approving edited livestock opportunities, whatever you want to call them, resources, has got to change. It's just crazy right now.
Yeah. Okay, well, we're almost at the top of the hour, so I think I'm going to close it off here. I want to thank Ed and Mark again for a very fascinating set of talks and bringing a lot of interesting ideas, lots of food for thought, and certainly a lot of things that we need to follow up on, and we will follow up on in this conference that I mentioned. Nicole has put the link for the registration to the conference in the chat, and also the link to the RFA, the AG2PI RFA that was released by NIFA a couple of weeks ago. So please have a look at those. Yeah, thanks all of you for attending. A couple of other things. We won't have a field day in May. The next one will be June 21. There are a couple of workshops coming up, one on water dynamics from molecular structure to phenotype, and then one on homomorphic encryption or to enable sharing of confidential data. So sign up for those. They usually fill up fairly quickly, so if you're interested, please sign up as soon as you can. And then of course the conference. So with that, thanks again to Nicole for all the logistics and Eddie for the technical side, and thanks to all of you for attending, and hope to see you in a couple of weeks or 2 months. Take care, everybody.